The novel coronavirus has infected millions, compelling people to stay away from loved ones and friends. Why is this virus so contagious, and what can we do to stop it? The answer lies in the virus's most potent weapon, spike proteins. Spike proteins play a key role in breaking into human epithelial cells so that the coronavirus can release its RNA, triggering an immune response called the COVID-19 disease. These surface proteins lie on a lipid membrane. The membrane is exactly why soap and water is such an excellent coronavirus killer, as it can destroy the lipids, leaving the RNA defenseless. The spike protein essentially creates a pathway between the virus and the cell. The exact viral entry mechanism is really complex, so I broke it down into a 3-step process to make it easier to understand. Step 1. Binding. The head of the protein comes in contact with the ACE2 receptor on the human cell, which helps regulate blood pressure. The spike is camouflaged with glucose, tricking the ACE2 receptor into believing it's a good cell. The receptor pulls and removes the binding domain from the spike protein. Sometimes the virus needs a little more help removing the binding domain. That's when a helper protein on the human cell comes along. It's called TMPRSS2, and it breaks the bonds between the two domains. This phenomenon is called proteolytic cleavage. From there, the receptor, binding domain, and helper protein all leave, initiating step 2, connection. The fusion domain now has a clear shot at entry, so it starts to elongate. It shoots a chain of amino acids across, called the fusion peptide, and it latches onto the human membrane. And now the third and final step, fusion. The fusion peptide, now acting as a tether, bends and pulls both membranes closer and closer until they touch and spontaneously fuse. Now the virus can enter. From there, it's pretty easy for the virus to damage the cell and continue its attack. So, what should we do to stop the virus? The coronavirus is at its most vulnerable when the spike protein is binding. That means stopping the spike protein from completing viral entry. If we can stop entry, we can stop infection. One vaccine solution uses the spike protein RNA sequence. The RNA is sent to our wondrous immune system so that it can develop antibodies specifically against spike proteins. Ultimately, these antibodies can stop the harmful spike protein from binding. No entry, no infection. This is just one type of mRNA vaccine, and it's currently being tested and shows great promise. The breakthrough here is we can utilize the properties of spike proteins to stop the coronavirus. Spike proteins are the problem, but they can also be the solution. Stay well.